Alright, so with this video I should cover everything that was cut off of that recording of the review session I did. So this is going to be the last lecture we had for this section, and it's going to cover long-term potentiation as it exists as a function of the hippocampus, or as it exists as a function of Nmda receptors. So when we're talking about long-term potentiation, he mentioned in class, long-term potentiation, four major features of long-term potentiation that you should know. So one of these is specificity. Specificity, what he wants you to get from this is that these synapses that perform long-term potentiation, or that have long-term potentiation type characteristics, have specific features. They have specific receptors that allow them to do this, right? And there's probably different types of long-term potentiation that exist in different parts of the brain or different parts of the hippocampus, but specificity is referring to the fact that you need certain things to do long-term potentiation. Duration, what he wants you to get out of this is that it's long-term, right? This isn't a short-term like facilitation or short-term depression. This is going to be a long-term effect on the neuron that's going to persist throughout, sometimes even throughout the lifetime of the organism. Here, I don't know why I'm not numbering them. Number 3, synaptic pairing. This is just the idea that synapses strengthen each other, right? Or I should say, the presynaptic and the postsynaptic portions of a synapse strengthen each other, right? So there's all kinds of signaling that goes on that causes the strengthening of a synapse. And that's all he wants you to get out of this third thing. And then finally, associativity. And this is one of the big colloquialisms that we hear in neuroscience. That's neurons that fire together, wire together. And the one I like, somebody told me, one of my professors, was that neurons that get drunk together, thunk together, or something like that. So however you want to remember that. But basically it's that neurons that fire at the same time or in the same place or anything like that are going to end up being connected in some capacity. And that's important for the fact that, during development, when you see an object, you want the neurons responsible for determining the form and the color and the shape and the movement of that object to all coincide in some sense so that you can get a more holistic picture of what that object is. That feature is all across neuroscience. So these are the 4 things he wants you to get from long-term potentiation. And we'll talk about the specific synaptic changes. But first, I want to talk about the basic structure of the hippocampus that he expects you to know. So if I'm drawing the hippocampus, we got kind of 3 major regions. So we have the Ca1 region, the Ca3 region, and the dentate gyrus. Ca stands for cornus ammonius, which in English means Ammon's horn, just because it kind of looks like a horn. Don't memorize that, but know that Ca is referring to this horn area. So that could be a good way for you to memorize the fact that Ca corresponds to the horn part. Not super important, but just helps me a little bit. So the cell populations that we care about exist in 3 locations. So what I'm going to draw here, this is going to be the dentate gyrus. These cells within the dentate gyrus are going to be known as the granule cells. So these are granule cells. And if you look up granule cells, there are also granule cells in the cerebellum, so don't get confused there. For some reason, they name them all the same sometimes. But these are going to project to the Ca3 region. More specifically, the Ca3 neurons, which are these pyramidal-shaped cells within that region of the hippocampal cortex, right? And they're going to be kind of shaped like that. So they interact with the dendrites of that. Actually, I drew that wrong. This part is actually going to be the dendrites. So they interact with the dendrites, and then the axons of the Ca3 neurons are going to project to the Ca1 neurons, So here's their little triangular shape. And then there's their axon. And then these are going to leave the hippocampus. So information coming into the hippocampus for synapses on these neurons in the dentate gyrus. And these are going to be, this is going to be your perforant pathway. So your perforant path is going to be the input to the hippocampus. This is going to be the output. And it has a lot of names. Sometimes it's called the fimbria. I don't think you should be expected to know that, though. But perforant path, you should remember. So these neurons, these Ca3 neurons, Ca1 neurons, these are going to be the ones that perform long-term potentiation. So this connection between Ca3 and Ca1 is where long-term potentiation exists. And so if I'm just going to point out a couple more structural features, this bundle of fibers is known as the mossy fibers. And then this bundle right here, right, likely there'd be a ton more because there's a bunch of Ca3 neurons. These are going to be the Schaefer collaterals. Right. So this is your general anatomy of the hippocampus. And if you ever have seen a picture of it, it's kind of that squiggly line looking thing. Basically, we just want you to get out of it the way these things are connected, right? You have your granule cells projecting to your Ca3 region, and I'll kind of outline these better. So here's your granule cells. They're going to be in your dentate gyrus. Just picking random colors here. Your Ca3 region is going to be like that, and then your Ca1 region is going to be like that. This is very general. We would never ask you to outline that, but that's just to show you that there is regional specificity there. But what we're going to do is we're going to zoom in on this right here. So we're going to take a closer look at that synapse, and what we're going to find is that these postsynaptic cells are embedded with something known as Nmda receptors. And what I'm going to do is actually move this like that. So this is your postsynaptic cell that I'm finishing drawing right here. And this is your presynaptic cell. These presynaptic cells are going to be glutamatergic, so they're going to release glutamate. And then from there, we know that they release glutamate, so I'm actually going to erase the presynaptic so we can zoom in on the postsynaptic. So, and this could have been drawn better. So here's our postsynaptic cell. It's kind of being flooded with glutamate. And glutamate binds to two different receptors on the postsynaptic cell. And we've talked about these in detail in the ionotropic channel lecture. But there's a couple features I want to go over again. So remember that AMPA, right? Remember that they always bind to two glutamate molecules. AMPA receptors, these are going to be selectively cationic, meaning they allow the influx of sodium and potassium. But there's one we're leaving out here, and for good reason. Nmda receptors are going to be non-selectively cationic. Non-selectively cationic. So the major difference here is that they're also going to lead to an influx of calcium. Calcium is the big important factor here. So if this is our AMPA receptor, this is our Nmda receptor. I'll draw it in a different color right here. So the major structural difference here is the fact that Nmda receptors have within them a magnesium ion that acts as a voltage sensor. Magnesium. So what's going to happen is even once this Nmda receptor is bound to glutamate and glycine, right? Because it needs glycine also to function. Even when it's bound to glutamate, it's not going to open unless that voltage sensor is removed. And the only way that voltage sensor is going to leave is if there's some reason for it to. If there's some repulsive force between the positive charge of the magnesium ion and some other positive charge. So what you get is you need sufficient depolarization into the cell via the AMPA receptor. So let's say, for example, here's my eyeball, and I'm looking at something and it just happens to be the color blue, right? So I'm looking at a picture of something and it's blue. That probably wasn't very smart, but for me to remember, let's say this is a really random object and just so happens to be blue. The cone photoreceptors in my eye that responds to blue best are going to be firing like crazy, right? And they're going to be eventually projecting to my hippocampus. Remember, this is the Ca1 neuron of the hippocampus. This is the postsynaptic neuron here. So if I just kind of glance at this blue color, I'm going to get a little bit of positive charge into this Ca1 neuron, but not much, right? There's not going to be much association between this neuron that's responding to the blue and the neuron that could be responsible for strengthening this connection. But let's say I concentrated on that color blue. So not only did I glance at it, I really looked at it and I really tried to like figure it out. What is that? What color is it? What shape is it? This AMPA receptor would probably respond or interact with a lot more glutamate and result in a lot more positive influx, right? So now this neuron responding to blue in the Ca1 region of the hippocampus is very excited. And what you end up getting is a buildup of positive charge that not only leads to more action potentials in the postsynaptic neuron, but it actually leads to this magnesium voltage sensor being kicked out, thanks to what we always talk about in this class, which is the repulsive forces between two like charges, right? So because this magnesium voltage sensor leaves, and I'll kind of erase it here, we now have an influx of both sodium, a little bit of efflux of potassium, but not much. But very importantly, we have an influx of calcium. And calcium is going to do something really specific. So calcium is going to lead to the activity of calcium calmodulin kinase. So this is calcium calmodulin kinase, which is going to lead to, as a kind of a eventual effect of this signaling cascade, is the exocytosis of vesicles that contain AMPA receptors, right? And we remember that calcium leads to vesicle exocytosis in a lot of different ways. So because it's leading to exocytosis of vesicles that contain AMPA receptors, let's draw this a little differently. So this is the postsynaptic neuron before. This is pre-long-term potentiation. Right, so long-term potentiation being this calcium leading to the increased exocytosis of these AMPA receptor vesicles. So this is pre-LTP. Let's say you have two ligand-gated ionotropic channels, so two AMPA receptors, and you get 4 molecules, or let's say 8 molecules, 4, 5, 6, 7, 8. 8 molecules of glutamate released. And this is just an example. Obviously, you'd usually have a lot more. But you could only open these two AMPA receptors and allow in a certain amount of current. But there's still glutamate hanging out in this synapse, right? So if you increase the exocytosis of AMPA receptors so that now, I'll draw them a little quicker because we're running out of time here, but now there's 4 receptors. Let's say, for example, now there's 4 receptors on this cell. Now every single molecule of glutamate that's released is bound to an AMPA receptor, meaning the same amount of glutamate release, right? So this is post LTP. In this case, the same amount of glutamate release leads to more current influx via these AMPA receptors. And that's all thanks to NMDA allowing calcium in, which leads to the exocytosis of vesicles that contain AMPA receptors. I hope that makes sense. Please let me know if you have any questions. Again, my email is drg1997@rams.collegestate.edu. And yeah, thanks.